Bismillahirrahmanirrahim. Assalamu alaikum, everyone. So today we are going to start the second module of Bio 101, which is about genes and development. In this particular module, we are going to learn about what is a gene, how it is linked to DNA, what is the relationship between DNA, genes, chromosomes, the words you have heard about in your O levels, A levels, or in last few weeks in this course. So in this particular module, what we are going to cover is how do we really know that DNA is the genetic material which is responsible for inheritance of traits. We call this as a hereditary molecule. How information is stored in the DNA, and then how this information is decoded in the form of RNA and proteins, etc. I'll try to upload all my lectures at least 48 to 72 hours ahead of our scheduled lecture time. And then in the 2 weekly scheduled times, we will discuss all the questions you have. You're expected to watch all these lectures ahead of time. Okay, so our today's lecture is it's the first part of the 2 lectures, what we call from genotype to phenotype, or also you can say DNA to phenotype. Phenotype is your appearance, how you look like, how a living organism look like. Here, for example, if you pay attention, here, this is strawberry, lemon, then you have these are I believe grapes, or there's some sort of berries. Then on this side you have carrots, different appearance. This variation, this diversity in the phenotype, the appearance, is actually encoded within the DNA of each living organism, each living species we have. So what I would like you to learn out of this lecture are following. So how do we know that DNA or proteins, which one of the two is actually the hereditary molecule? In the beginning of last century, one of the fundamental questions life scientists were working on, that whether it is DNA or protein which is responsible for inheritance of traits from parents to offsprings. So from today's lecture, we are going to learn this. We are also going to learn how information is stored within DNA, and then how DNA is copied. You should know that when a living organism, it grows, let's say bacteria, from one cell, it divides into 2 cells. All the DNA which is stored in mother cell is duplicated, is copied. We call this process DNA replication. And this DNA is then passed on to the daughter cells, the offsprings. So how this process of DNA replication takes place, we are going to learn from today's lecture as well. And then in next lecture, which will be the second part, today is the first part, in the second part, we are going to learn how the information encoded within the DNA is decoded, how the DNA, which is just 4 alphabets A, T, C, G, in random orders, apparently random orders, but how they make a specific code which gets decoded in the form of messenger RNA, then a protein. And we see this kind of phenotype, the appearance. This is due to decoding of the DNA of strawberry, decoding of the DNA of lime, or this berry, or carrots. So we are in the second part, which will be on Friday. We will learn about this decoding of information, and that is called central dogma of molecular biology. So today we are going to start learning these three particular concepts. So as I said, and in the last few lectures, you have already learned that within the living cells, the information, the genetic information, is stored in the form of DNA. This is a bacterial cell. This is prokaryote. This is a eukaryotic cell. You can see in eukaryote, there are membrane-bound organelles. So this is the nucleus, for example. In prokaryotes, there is no membrane-bound nucleus. The genetic material, the DNA, is just bound to cell membrane, and it's like a one big circular molecule in bacteria. In case of eukaryotes, within the nucleus, we have these structures which we refer to them as chromosomes. DNA, in case of a eukaryote, is packaged together with specialized proteins called histones. There are 4 kinds of histones: H2A, H2B, H3, and H4. 2 molecules of each of these proteins, which make a histone octamer. We draw it like this: 2 molecules of each, H2A, H2B, H3, H4. They make a pulley-like structure, and then DNA is wrapped around this in 2 circles. It goes like this, and 146 base pairs of DNA around this histone octamer. This makes fundamental unit of eukaryotic chromosome, which is called nucleosome. Nucleosome. Histones plus 146 base pair of DNA, they constitute a nucleosome. The nucleosomes are then, just a moment. So nucleosomes are the fundamental unit of chromosomes in eukaryotes. This is, for example, a single nucleosome. So these are histones coming together. This is a single nucleosome, 146 base pairs plus histones, next nucleosome, and you can see this nucleosomal packaging of DNA in the end results in the form of a chromosome. So you understand the relationship between DNA and chromosomes now. Chromosome is one complete unit of hereditary, which is inherited from parents to offsprings. And chromosomes are then further in eukaryotes are based on their structure. So this is called centromeric region. The termini are called telomeres. And then different lengths of DNA, and we will learn about those lengths or specific codes, are called genes. A single gene, for example, a tiny part of DNA here will be a one gene, then another gene, independent genes. Okay, there could be hundreds and thousands of genes on the single chromosome. For example, if we consider this as our cell, a human cell, there will be 46 chromosomes here. Okay, normally chromosomes don't look like this in a normal cell. This structure is visible when cell undergoes mitosis, undergoes cell division. In contrast to eukaryotic cell, in prokaryote, we don't have such nucleosomal packaging. We don't have independent chromosomes. In bacteria, we have one big circular molecule of DNA or chromosome. This is bacterial chromosome. One big circular chromosome, which is attached, and we will learn about bacterial chromosome. But what is important, whether you are a eukaryotic cell or a prokaryotic cell, the genetic material within a living cell comprises of DNA, and you have already learned about DNA. In prokaryotes, there are other DNA molecules as well, which we call plasmids. And plasmids are extra chromosomal. They are besides this chromosomal DNA, which is present in the bacteria. And these plasmids, they have their independent origin of replication. We will talk about what is role of origin of replication when we are going to talk about DNA replication. I told you DNA replication is when this plasmid is going to replicate itself and result in two identical molecules like that. So these are extra chromosomal molecules, and they have their independent origin of replication. They don't rely on or depend on the chromosomal DNA. Okay, they have their own, and I'm talking about plasmid. Okay, plasmids usually have antibiotic resistance genes on the bacteria harbor antibiotic resistance genes on plasmids. Now, this is a map of E. coli, simple bacteria, and genome. Genome is the word you see. Genome is all the genes present in a cell. It can be bacterial cell, it can be human cell, it can be mouse cell, etc. So this is a circular chromosome, nucleotide number 1, going all the way like this. It's 5.4 million base pairs. Okay, E. coli genome. And each green line or red line, these are basically representing independent genes, which means, you know, a specific segment. So what is a gene? If I linearize the complete E. coli genome. What is genome? I told you, basically your DNA sequence is your genome. A, T, G, C, A, T, C, whatever, you know, these nucleotide sequences. Now there's a specific reading frame, specific code, which constitutes a gene. And I'll talk about this in detail. So this will be one gene, which will, when it will be decoded, it will encode a specific protein. So protein coding unit of DNA, is called a gene. Then there will be next gene, another gene, and a coordinated action of genes, they enable a living cell or life to survive and live normally. Now, you already know that there are 4 nitrogenous bases. So A always pairs with T, G always pairs with C. So guanine always pairs with cytosine, adenine always pairs with thymine in DNA. And they are paired, they are bound through hydrogen bonds. So guanine and cytosine makes 3 hydrogen bonds, adenine and thymine makes 2 hydrogen bonds. In DNA, whether you are a bacterial DNA or a prokaryotic DNA or a eukaryotic DNA, it comes in a double-stranded form and in a helical structure. This is the helix, you can see. So this is the helix. There are 10 base pairs in each turn. So if this is from here to here is one turn, there will be 10 nucleotides. Okay. The red ribbon here is referred to as sugar phosphate. We call this backbone. And then the nitrogenous bases, they come within. And this double helical structure is actually has a very, very specific conformation. On the one side, it has this major groove, and on the other side, it has minor groove. And these major and minor grooves, they provide a very, very specialized sites for proteins to bind and then regulate the expression of different genes. This helical structures, if we try to see the size of the DNA present in a human cell, let's say, if we take all the DNA on all these chromosomes, we linearize this DNA in a single cell, I'm talking about, the length is 2 meters. And the size of the nucleus is 10 micron. So you cannot package 2 meters long thread in a nucleus of 10 micron size. So this nucleosomal packaging and this helical structure of DNA, which then, gets supercoiled. It not only is like this is a linear, it folds back, and then, in the form of nucleosomal DNA or, packaging, it becomes like a dot. If you take linearized DNA, it's just like you have to package a grapefruit within a lemon. Lemon is the nucleus and grapefruit is the DNA size. So this nucleosomal packaging and this helical structure, which then becomes superhelical, supercoiling. An easy way to understand is if you take a rubber band and you twist a rubber band from both sides. Initially, it will be linear. You keep twisting, and then it will fold back on top of itself. It will keep folding as you keep twisting, and then it will become a dot. So this nucleosomal packaging, then this helical structure of DNA, all this is very, very important for packaging of DNA inside a nucleus, which is much smaller, which is of much smaller size. Now, let me recap what I have just told you. What we have learned: DNA, which is, let's say, A, T, G, C, A, T, C, A, G, G, A, C, just a random sequence I'm writing. It's complementary sequence you can imagine here. Okay. Just imagine like this. What I have told you in eukaryotes, DNA 146 base pairs plus H2A, H2B, H3, H4, 2 molecules of each. They make a pulley. 146 base pair of DNA goes in 2 turns like this. Then next nucleosome, DNA goes like this. Then next nucleosome, DNA goes like this. Then next nucleosome. So one single 146 base pair of DNA plus histones is called nucleosome in eukaryotes. And these nucleosomes joined together like this, we call them chromatin. And eventually I showed you that such a chromatin or chromatinized DNA, it is visible as a chromosome in a nucleus. If we have 46 chromosomes, we'll have 46 such structures. Now, what is the relationship between DNA, gene, chromosome? So we learned what is relationship between DNA and chromosome. Where does gene come in? A specific protein coding region, for example, here, each nucleotide we know, and three nucleotides constitute a genetic code which encodes an amino acid. So a specific code which result in formation of a specific protein, that length of DNA is called gene. So DNA, now you understand DNA, gene, and chromosomes. Okay? We'll go through this concept again and again. Now, each living organism has its unique genome, unique DNA. Our genome is, human genome is very similar, like 99% similar to mouse. But we are very different. There is, although the DNA sequence and the genes are similar, but we grow different. This diversity in different species is actually encoded in our DNA. If we just look within the bacteria, so these are bacteria, for example, cocci, bacilli, they come in. So these are cocci are, for example, diplococci, which causes pneumonia, streptococcus pneumonia. It's round form. Then they make such structure. Bacilli are rod shaped. Then there are spiral shaped bacteria. Now, how they become different? They become different because they have different DNA. Their genetic code is different. Their genes are different. I just showed you E. coli. E. coli have 5 point some million base pairs. E. coli is also a bacteria. So look at the diversity, the variation. This variation within different species, between different species, is actually due to variation in the genome. Difference in the genome. Yeah, so here it is. Variation in the genotypes dictates variations in the phenotypes. Phenotype, I told you, when we'll talk about genetics, these two words will become even more clear. Phenotype is your appearance, and genotype is specific combination of genes. Now look at this. This is citrus. This is same species, citrus. But look, they are different. You have these red bloods, you have oranges, you have these what we know as the loose skin, which is quinoa, which is this one. I think they are called mandarins, and these are the oranges. Now, these are carrots, but they contain specific combination of different genes. We'll talk about them, which are called alleles, etc. So, specific combination of different genes is referred to as genotype. Your genome is different, and that genome dictates your appearance. Look, corn. We'll talk about all this in great detail in our genetics lectures, which will be next week. Now, yeah, so talking about variation, look again, you have, variation within our eye color, blue eyes, brown eyes, black, green, etc. Hair color, hair structure, curly hairs, extremely curled, straight, etc. All this is dictated by genes. These different traits, or characters, skin color, etc., they are controlled by specific combination of different alleles or genes. We'll talk about all this in our genetics lecture. What is important for you today is to learn that the differences we see between different species and then within species is actually encoded within our DNA, is present within our DNA. So we start our life from very, whether we are a mouse, human, plant, cactus, or a flowering plant, we all look same on day one. Single cell, sperm and oocyte fertilize, and we are single cell. Our beginning is very simple, but then based on the genetic code, our genome, our chromosomes, what we harbor within our cells, we become very different. A complex phenotypes we develop. I'm trying to re-emphasize again and again that the DNA we have in the nucleus, it contains very specialized information, which not only ensures that we develop the way our parents look like, okay, and the information within the DNA makes us different from each other as well, the different living organisms. Now, how, if information is stored in DNA, how this information is making us different? This information in the DNA is decoded in the form of proteins. So from DNA, a molecule is decoded, which is called mRNA, and this is decoded in the form of a protein. All the collection of proteins in my single cell, all the collection of proteins in a single cell. So I have 46 chromosomes in each cell in my body. My body comprises of billions of cells, and each cell contains 46 chromosomes. Each chromosome contains hundreds and thousands of genes. Each gene encodes a protein. So all the collection of proteins present within my cell is referred to as proteome. And proteins are basically the functional molecules. DNA is the software as well as the hardware, but this DNA is decoded in the form of a real functional molecule, which is the real hardware of a cell, and we call them protein. All the proteins within the cell are called proteome. So we had two words. Previously we used word genome. What was genome? Collection of all the genes in a cell. The genes, they are decoded to proteins, and collection of proteins are called proteome. And let's say this is one protein, this is another. These are unique proteins coming from unique genes in my chromosomes. Based on your proteome, you can classify living organisms. For example, this branch of tree is bacteria. This branch of tree is eukaryotes. This branch of tree, this is tree of life. Okay, this is tree of life, and these branches, they are telling which organism is how many million years apart in the process of evolution from each other, how much different we are. Okay, we call this study phylogenetic tree, like that. This is for example, if you pay attention here, this is mouse, mus musculus. Then this is rat, ratus, Ratus norvegicus, and then this is human, Homo sapiens. Okay, this is Homo sapien branch. This is mouse and rat, but still very different. And if you look at their evolutionary distance, it will be millions of years. Why they are different? How can we draw such a tree of life? Is basically based on the genome sequence or the proteins they encode. Now, in the beginning of last century, so this was all about a refresher or a intro about DNA, link with chromosomes, genes, proteins, etc. We'll go through them again and again in these 8 lectures, which I'm going to teach you. So now let's talk about how do we know that DNA is actually the hereditary molecule. Hereditary molecule means the molecule which contains information which dictates inheritance of traits. The molecule which ensures that the offsprings, they look exactly like the parents. Bacterial cell, it produces bacterial cells. Humans, it produces babies. Cats, it produces kittens. Dogs, they produce pups. So we, in the beginning of last century, one of the fundamental question was whether it is DNA or protein. You will be laughing because, you know, today, you know, proteins are encoded by DNA, they are same. But in the beginning of last century, it was not clear. People working with proteins, they were saying protein is independent entity and, you know, it controls hereditary. Then the people working with DNA, they were saying, no, it's the DNA molecule. So one of the finest experiment, which was done by Hershey and Chase, they solved this enigma. And Hershey and Chase, they both did a very simple experiment. They used bacteriophage. Bacteriophages are viruses, as the word indicates. So they infect bacteria. Phages are viruses. Viruses which infect bacteria. They eat up bacteria. This is the electron micrograph. This is a real picture. This is how this virus looks like. And its structure is drawn here. It has, you know, head, it has neck, then this collar, and then this tail. All this head, neck, collar, tail, all of them are made up of proteins. The DNA of this virus is present within this head. This blue one is the DNA. Now, this virus, this is the real picture, electron micrograph. This is how it looks like. Look here, there is also electron micrograph, a real picture. The bacteriophage is attached to bacterial cell. This is bacteria. And then what it does, it injects its DNA within the bacteria. When it goes within the bacterial cell, then it makes many more components, which are head, neck, collar, tail, etc. New viral particles are assembled and they burst open. They kill the cell, release, go to infect the next cell. Now the question was, Hershey and Chase asked, okay, whether the proteins or the DNA within the bacteriophage, which one of the two, proteins or DNA, are the hereditary molecule? Simple system they used. What they did, they came up with a very intelligent, very smart approach, and they said, okay, we are going to use bacteriophage T2. This is the type of the phage. We are going to grow them in the presence of radioactive sulfur, S35, radioisotope, or we are going to grow viruses in the presence of radioactive phosphorus, P32. Why they came up with this idea? It's very important. It decides that it contains the crux of this whole experiment. Why they are growing viruses in the presence of sulfur or phosphorus? They knew that amino acids, methionine or cysteine, you know, amino acids are the fundamental blocks of proteins. Proteins are made up of 20 amino acids. Now, two of these amino acids, methionine or cysteines, they contain sulfur. And if we will grow a phage in the presence of S35, that phage will contain proteins which will be radio labeled, S35 labeled. Okay. And if we grow phages in the presence of P32, we know DNA has phosphate backbone. DNA contains here phosphate backbone. So phosphate is essential component in the synthesis of DNA. So when we will grow viruses in the presence of P32, the DNA will incorporate this P32. Now, what they did, they set up two experiments side by side. In one, they mix bacteria with viruses which were grown in the presence of radio-labeled phosphate. And the other experiment was they mix bacteria with viruses which were grown in the presence of S35, radio-labeled sulfur. In this case, we know sulfur is component of amino acids, so the proteins of virus will be radio-labeled. And here, DNA, which is this one, will be P32 labeled. Phage will infect this bacteria. Okay. After infection, what they did, they simply spun it down in maximum speed. What they waited for after some time. So bacteria was attached by viruses. Whatever material they inject, it went into the bacteria. They spun it down in a blender so that they become separate. Bacteria and viruses become separate. And whatever material phages have injected, they remain within the bacteria. Now, it was followed by high-speed centrifugation. They now spun the whatever material was here in specialized tubes. They use centrifugation machines, and then under centrifugal tube, what they wanted to see where the radioactivity is present, P32 or S35. Viruses are very, very small molecules. Under this kind of centrifugation which we use, we cannot settle them down. But the bacteria are heavier molecules. Bacteria are heavy molecules, and due to the centrifugal force, they will settle down in the form of pellet. So all the bacteria, after this centrifugal force applied on them in centrifugation machine, all the bacteria will settle down, and we will have liquid. So this liquid is called, phase is called supernatant, and the one which settled down is called pellet. Okay. Now what they wanted to see, where is P32 and where is S35? What they discovered that pellet contain all the P32, which means bacteria contain all the P32. And if bacteria contains all the P32, it means it's the DNA of phage which is injected within the cell, bacterial cells, because radioactive sulfur, it remained in the liquid phase or supernatant phase. It did not go within the pellet, within the bacteria, which means phage actually injects its DNA, and DNA is the molecule which contains all the information which results in synthesis of new head proteins, tail proteins, collar, and then, a new offspring or new viral particle is synthesized. Okay. So this experiment solved a long-standing question whether it is the protein or it is the DNA. And they demonstrated that it's the DNA which contains all the information which is required to make more copies of T2. It's just like a bacteria. Analogous conclusion can be that all the information within the bacteria which is required to make 2 new bacterial cells is encoded within the DNA. So it's the DNA which is hereditary molecule. Okay. Now, how is this information stored in the DNA? I think in the first module you talked about nucleotides, you talked about nitrogenous bases. I'm just going to go through quickly what is this information, how this information is stored within the DNA. So we have learned one thing, DNA is the hereditary molecule. And Hershey and Chase did a beautiful experiment to prove this concept. Now, how this information, because DNA is not, in the beginning I said it's just random, A, C, A, G, A, G, C, A, T, T, A, it, for a layman, it looks a random sequence, You pick up mouse gene sequence, you pick up human, bacteria, you won't be able to differentiate because they will all look same. But we know in 21st century that there is a very, very specific information in bacterial DNA, in human DNA, in mouse DNA. How this information is stored, the storage is same from bacteria to humans. The code, the genetic code is same. So again, as I said, one of the strong points in the theory of evolution is that we all contain this genetic code. We all contain this DNA. The code which is decoded in the form of protein, it comprises of this, what we call triplet code, 3 nucleotide code. Otherwise, it's random sequence. Okay. Now, how this information is unique? Because if it is not unique, then we cannot grow into complex, unique phenotypes. This information is indeed unique. So we humans, we have 46 chromosomes. Okay. Each chromosome, it exists in pairs. Okay. When chromosomes exist in pairs, so our human cell, which contain 46 chromosomes, this cell with 46 chromosomes is called diploid, and it's represented or written as 2n, diploid. It means there is half of 2n, which is 1n as well. It's called haploid. Our haploid cells are egg cell, oocyte, or sperm cell, the male reproductive cell and female reproductive cell. They fuse together through the process of fertilization to make a diploid cell. This diploid cell divides into further diploid cells, and we eventually grow as beautiful babies. Okay. Now, chromosomes, what are this diploid and haploid? When chromosomes exist in pairs, cells which contain each chromosome existing in pair, for example, this is chromosome 1 and its exact identical copy, chromosome 1, chromosome 2 and its exact identical copy. So these identical copies of same chromosomes are called homolog chromosomes. Homolog. This is homolog of chromosome 2, homolog of chromosome 3, homolog of chromosome 4. That's how we are 46 chromosomes. The number of nitrogenous bases, the number of A, T, C, G, the nucleotides, they are 6.4 billion base pairs in each cell. In a single cell, 46 chromosomes, they are made up of 6.4 billion base pairs. This is where our life starts. Sperm, a haploid cell, a haploid oocyte, fertilize, it becomes now 2n, it starts dividing mitotically. We'll talk about mitosis as well. 2n, 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 and we grow like this. Okay. Mouse has its own number of chromosomes. Different plants have their own number of chromosomes, but our genetic information is unique. The DNA sequence is unique. The information, this unique information in each species is stored, as we learned, in the form of DNA. And Watson and Crick in 1952, they solved the structure of the DNA. They discovered this hydrogen pairing based on the X-ray crystallographic structure of DNA molecule. And they said that guanine is pairing with cytosine, adenine with thymine, and DNA is a helical structure. There are major grooves, there are minor grooves. Now we can pinpoint through DNA structure, we can pinpoint each atom present in DNA. Okay, where is oxygen, where is hydrogen, sugar phosphate backbone. If you have the structure of DNA, you know at the atomic level the whole structure of DNA. This was a big discovery, because this double-stranded DNA, structure of this double-stranded DNA in the form of helix, it turned out that actually DNA and protein, they are not different. DNA encodes protein. This specific combination ACGTCT, it actually encodes. So each triplet code encodes specific amino acid. And amino acids, they are the building blocks of proteins. So what we were thinking of in the beginning of last century, that proteins are independent molecules, they have no relationship with DNA. It turned out that DNA contains all the information. The nucleotides of this DNA is not a random sequence. It is a very, very specialized code. And this code is decoded in the form of messenger RNA, which is then decoded in the form of protein. And that is the information stored, how it is stored in the DNA. If you look at if we try to, look at the structure of DNA, what we learned that DNA comes into anti-parallel strands. Anti-parallel means you have 5' to 3', and then the anti-parallel strand is 5' to 3'. So we write them like this: 5' to 3', and the other strand will be 5' to 3'. This anti-parallel strand structure of the DNA is actually the reason which makes the hydrogen pairing between different nitrogenous bases possible, and also contributes to this helical structure. So what is 5'? So this is, for example, this one is nucleotide which encodes which has thymine as a nitrogenous base. Nucleotide is what? Nucleotide is phosphate group, deoxyribose sugar, and nitrogenous base, in this case thymine. One nucleotide is, so phosphate group is on the 5th carbon, but the extension of DNA is always at the 3' end, because at the 3' end there is a hydroxyl group. Okay, so this hydroxyl group binds to phosphate group of next nucleotide, and it makes a phosphodiester bond. This is called phosphodiester bond. Through phosphate groups making this phosphodiester bond with hydroxyl group of this nucleotide, where, so what we have here hydroxyl group, we have here phosphate group. if we have to elongate chain of DNA, the next nucleotide will come here. Okay, which was 5 position of a nucleotide, which is normally phosphate is on the 5th position. Okay, here the hydroxyl group will be free. So this is 3' end. So here you have 5' end, and then the free 3' hydroxyl here is that's why we write 5 to 3, and in anti-parallel 5 to 3. You cannot have 2 5 like this. It's not possible. DNA strands will not align. They will not make base pairing. It's simply not possible. Okay. In one helical turn, starting from here to here, you have 10 nucleotides. Okay. So there are 2 hydrogen bonds and in adenine and thymine, and 3 hydrogen bonds in guanine and cytosine. So guanine and cytosines are called purines. No, sorry, not guanine and cytosine. Adenine and guanine are purines, and cytosine and thymine are called pyrimidines. Okay. So how this information, we said information is stored in DNA. This is the structure of DNA, or how DNA elongation takes place. Now, what is the information then there? Let me draw here information. We have seen imagine CGA this is a double-stranded DNA. Each nucleotide, each of these 3 nucleotides makes a code. We call this triplet code. Triplet, whether you're a bacterial cell or a human cell. Now, each of this triplet, it's also called a codon. Codon from the code. It's a triplet code. So for example, ATG, when it will be decoded, it will be responsible for making an amino acid called methionine. CAG is a code for glutamine. Methionine is M. Q is symbol of amino acid called glutamine, and so on. So this is the code or storage of information for protein synthesis, and proteins are functional molecules. From our hair to our nails of toe, we are made of proteins. We'll talk about this, all this in next lecture. Now, I told you in the beginning, it is very important for a living cell that it is able to divide and pass on whatever DNA it has to the daughter strands. We call this information, this process, DNA replication. For a living cell, like we saw here, we said, after fertilization, this is very important concept. After fertilization, we become a diploid cell, and this diploid cell divides into 2 daughter cells. Each cell then keeps dividing into 2 daughter cells, and then eventually we start becoming, structure, organs, and etc. We'll talk about different stages of development in last 2 lectures. But what is important here? What is happening when a cell is dividing? Before it divides into 2 daughter cells, it should replicate. It should amplify all the DNA which is present in the form of chromosomes, all the DNA on these chromosomes. So 46 chromosomes, they have to be duplicated into 92 here so that each of the daughter cell becomes, each of the daughter cells, it inherits 46 chromosomes. So we call this process when DNA copies itself and replicates. This process is called DNA replication. How DNA is copied. This is very important. And then in next lecture, we will learn about how information is decoded. Today, we are going to learn about how DNA is copied, how DNA replication takes place. Why it is important. I told you why it is important. You can't grow as a living organism if DNA replication does not take place. DNA replication is at the heart of cell division, mitosis. Okay, so you have DNA molecule. This arrow means it can replicate. I just explained you why replication is important. In order for a cell to grow, it has to decode its DNA. And that process, decoding of DNA into a molecule called RNA, is this process called transcription. RNA is decoded into protein, is called translation. Each living organism has its own unique genome and proteome, and then we grow as plants, mouse, fish, Einstein, blah, blah, blah. Okay. And our variation within the genome also encodes different traits. That we'll talk about this when we learn genetics. However, it is very important. We learn this flow of information from DNA to RNA to protein, transcription and translation. We call this central dogma of molecular biology. How DNA replication takes place? Today's lecture will contain this information. We'll learn this concept. There are enzymes which are called DNA polymerase, which ensures that DNA molecule is copied. So this DNA, if this is one molecule, 2 molecules of DNA are made, and the enzyme which does this is called DNA polymerase, and the process is called replication. Enzyme which decodes DNA into RNA is called RNA polymerase, and the protein machinery which decodes RNA and synthesizes protein is called ribosome, and the process is called translation. However, there are some exceptions to this central dogma, and that is RNA. There are organisms and viruses which break down this central dogma. For example, COVID is an RNA virus. It does not have DNA, and it can synthesize proteins, and its genome comprised of RNA. Many viruses, hepatitis C as well. Hepatitis C virus is also an RNA virus similar to COVID, plus strand RNA viruses. But there are also viruses like AIDS viruses, HIV virus, which have RNA. It goes back to DNA, and then it has a DNA intermediate in their life, goes back to RNA, and then protein. So there are exceptions to central dogma as well. That's a very interesting study, that very interesting field which proposed that RNA was the primitive molecule in our evolution. But that's all a hypothesis. But then I wanted to bring it to your knowledge that there are exceptions to this central dogma as well. Now, why DNA replication is important? I told you when a cell divides, when a cell decides that I have to divide into 2 cells, and I gave you example of our own beginning of life from after fertilization, the cell must replicate all the DNA on its chromosomes into independent copies. And then, so for example, here it's a dividing cell. It has multiplied, it has amplified all its DNA. It has copied all its DNA. And then you can see it is dividing into two cells. Now these are two daughter cells. If there were, let's say, 6 chromosomes in daughter cell, in mother cell, by this stage it has already 6 here, so it means there were 12. It replicated. It's all DNA, the chromosomes, and then each of the daughter cells, it has 6 chromosomes. It has inherited 6 chromosomes as well. How this DNA replication takes place, it's very important. As we already know, DNA, synthesis of DNA, and I just want to re-emphasize again, synthesis of DNA or RNA, it always takes place in 5-3 direction. But protein synthesis is always from amino terminal to carboxy terminal, the peptide bond. Peptide bond is always, so this is your ribose sugar. This is the deoxyribose, the sugar which is present in DNA. This is the sugar, ribose sugar, which is present in RNA. You see 2 hydroxyl groups. In deoxy, you don't have, here you just have a hydrogen atom. And we already saw why 5-3 elongation of DNA takes place. In case of protein synthesis, the new amino acid, when ribosomes are going to elongate and make a peptide chain, here the new peptide bond will be formed. And in protein synthesis, we say it's from amino to carboxy terminal protein elongation or peptide formation takes place. Now, Measles and Stahl in the middle of last century, they discovered, once the DNA structure was discovered, the question was how DNA actually replicates. And the conclusion of their experiment was, I would like you to go to chapter 11 and read this experiment in detail. And the conclusion was, they said that the DNA replication is semi-conservative. Semi-conservative means that when the mother DNA or original DNA is going to replicate, the 2 strands will separate completely and copy strand, the new strands, which are these red ones, they are synthesized, the daughter strands, and that's how DNA is replicated. In contrast to semi-conservative, the other models which people were discussing were conservative. Conservative means, you have one copy, it will remain as such, and then the new copy will be formed. Or dispersive means, you have the parental strands, both the parental strands, and then in between you have, new nucleotides formed. But Measles and Stahl, the experiment they showed, they said that these 2 models are wrong, and today there is consensus. We all know that DNA replication takes place following semi-conservative DNA replication model. Now, what is semi-conservative model of DNA replication is? This is just again, explaining you 5' to 3' elongation. When you have a new nucleotide is coming, you can see here you have the 5' and the phosphate, triphosphate here, which will make chemical bond, phosphodiester bond with this hydroxyl group, and you will have cytosine making 3 hydrogen bonds with guanine, and this phosphodiester bond will be synthesized, and you have 5-3 chain elongation. Who does this? The enzyme which performs this function of addition of nucleotides. So here, the next nucleotide will be which one? The next one will be here. Guanine will come, next nucleotide. Okay. So enzyme which performs this function is called DNA polymerase 3. DNA polymerase, it can copy DNA into a DNA. Now, the important question in case of DNA replication is how DNA replication starts. We said DNA polymerase can add a new nucleotide. But let's say I have this DNA molecule. I have to copy this. I separated this. The hydrogen bonds are broken. Okay. Now, DNA polymerase 3, it adds nucleotides to free hydroxyl group. Where is free hydroxyl group? This is 5 prime, this is 3 prime, this is 5 prime, this is 3 prime. So if I have to start DNA replication here or here or here, let's say, because DNA replication goes 5 to 3, where is the free hydroxyl group? We don't have hydroxyl group. So living cell is very intelligent. It's one of the best engineering machine. It has an enzyme which is called primase. And this primase, it synthesizes an RNA primer. Complementary, so you have two strands separated now. If we have to replicate, make copy of this DNA, what it does, primase adds a RNA primer, RNA molecule, small, few nucleotides, complementary to this DNA. What it provides a 3 prime hydroxyl group. Now, this 3 prime hydroxyl group is recognized by DNA polymerase 3, and it starts chain elongation, and you see new DNA is formed, exact copy of this one. Okay, the other strand of this will also be copied similarly. Now, a question which must be coming to your mind, if we have 5 prime to 3 prime and 5 prime to 3 prime, if we are going to copy this one 5 to 3 and DNA is being zipped apart here at this point, this is the direction in which DNA is being zipped apart into two different strands like this. Okay, the question is, how can we synthesize here 5 to 3? It's not possible. We have to go into this direction. So fundamental concept in DNA replication comes in here, that one of the two strands replicates continuously. We call this one, which is without any disruption, perfect anti-parallel, 5 to 3 and 5 to 3. This is continuously replicating strand, and the other one is discontinuous. What is discontinuous? Discontinuous means it will be in bits and pieces. What RNA primase does, RNA primase, on this side it's simple, continuous replication. DNA is being zipped apart and DNA polymerase 3 keeps adding now, once RNA primase added primer here. But on the discontinuous strand, or what we also call lagging strand, RNA primase adds a small primer. When more DNA is available, it will add another primer here. When more DNA will be naked, it will be, and then DNA polymerase will extend this direction 5 to 3, 5 to 3. Okay, and you have now many such short fragments like this. So that's why we call this discontinuous. And these short fragments are called Okazaki fragments. Okazaki fragments. Once DNA replication is complete, then DNA, so this is how it will look like before I go to the next step. So this is a continuous strand. Okay, DNA polymerase 3 keeps adding. Here, when based on the availability of DNA, so RNA primer comes in and DNA polymerase 3 goes into this direction, because this is the lagging strand. The helicase at this place is called DNA replication fork. This is the point where DNA is being zipped apart. Okay, and we have helicase is the enzyme which unwinds the DNA. Hydrogen bonds are broken. DNA becomes single-stranded. Primase comes in. It adds this RNA primer, and then DNA polymerase comes in and elongates DNA into this direction. Okay. Now, you must be wondering, what is the role of RNA molecule in a DNA? What is the fate of this RNA molecule? So what happens once DNA replication is complete, the and you still have these RNA molecules within it. DNA polymerase 3 has made all the red ones at the DNA strands. DNA polymerase 1, another enzyme, a different enzyme comes in, it hydrolyzes the RNA primer, degrades, okay? And then these RNA primers are replaced by DNA molecule, okay? No more RNA primer is there. And this is followed by another enzyme which is called DNA ligase. Ligase then connects this DNA and the next strand together. No gaps. Ligase is the glue. And then you have a perfect copy of parental DNA. There's another concept called proofreading. DNA proofreading, just like you give your document to some of your colleagues to please read my document if I have made some mistakes, some spelling or typos, etc. So proofreading means the DNA polymerase, when it adds, let's say there is an A there, DNA polymerase 3 is going to add T here. If there's G there, it will add C there. If there's C there, it is going to add G there. Sometimes 1 in a million, it can create a mismatch. The DNA polymerase can, by mistake, instead of C, it can add T here. But enzyme has proofreading activity itself. It goes back and it reads, hey, I made a mistake. And this enzyme then removes this T, replaces with G. We call this proofreading activity. Okay? There are other mechanisms as well. Even after DNA replication is complete, there's a scanning mechanism which scans that there's no major mistake in the genome. And let's say if there is a mismatch during the DNA replication, the mismatch, we call those proteins mismatch repair proteins. They're going to come and they're going to excise the mismatch base, just the base, cytosine, or they're going to excise the mismatch base only, the phosphate backbone and the sugar is there, and they're going to replace with the correct base. There's another one which is called excision repair. In this case, if there's a mismatch, the repair machinery, what it does, they come in and they remove a small chunk of nucleotides, a fragment which contains this mismatch, and DNA polymerase comes, and then DNA polymerase again synthesize the correct bases. This is basically telling you the complexity of DNA replication, how complex it can be. Imagine I told you that the DNA is in eukaryotes, for example, it's chromatinized, it's in the nucleosomes, DNA replication machinery, and then DNA is supercoiled. For example, this is, the circular chromosome of bacteria, for example. When it starts from here, origin of replication means the point. Origin of replication is the point where the double-stranded DNA will be zipped apart. Hydrogen bonds will be broken. The rest of the DNA is still circular like this. But here it is broken, and now RNA primase comes and starts replicating. It starts making the complementary, you see the copy DNA, and then it has to, resolve these structures because there are coils coming in. DNA is being zipped apart, okay? And when you are zipping apart DNA, there are more supercoils are being generated ahead of the replication fork. There's an enzyme called topoisomerase which resolve these supercoils. There is a beautiful mechanism. It creates a small nick, resolve these entangled parts of DNA, and then resolve two beautiful DNA molecules, daughter molecules, okay? Parental and then two daughter, you see. If you think of, chromosomal DNA, in our case, it's not circular. So chromosomal DNA has many origin of replication. Bacterial DNA has single origin of replication. Chromosomal DNA has to have many origin of replication because we have 46 chromosomes. We have 6.4 billion base pairs in a diploid cell to be duplicated within a span of few hours. And it's not possible to have just one origin of replication in nucleosomes. So there are multiple origin of replications. It replicates in this direction, in this direction. You can imagine sometimes you'll have continuous strand, discontinuous, based on which of the two strands are being replicated and which direction of the replication fork is. And then they may, this is how long chromosomal DNA is replicated. And we also have top isomerases there as well. We'll talk about polymerase chain reaction in our BIO100. Just read about this. What polymerase chain reaction is. This is basically PCR is the machine. You in last 1 year you must have heard about, PCR for COVID is positive, PCR for COVID is negative. PCR is a machine which mimics the process of DNA replication, this one, and you can, using PCR, you can copy, let's say you have 100 copies of, for example, COVID in your genome, in your blood sample. You can amplify after following this DNA replication principle. This machine is designed. You can amplify exponentially this 100 after 35 cycle. You can think how many millions you will have, and that's all. Few molecules of DNA can be detected in blood samples, from our hair samples, from cheek cells, from our in forensics, in criminology, to solve crime cases. PCI is also used to amplify from the toothbrush when they have to resolve in case of accidents, the dead bodies are burned. They say, okay, give us toothbrush of the deceased, and they try to amplify from that toothbrush because there are some cells which are coming from our cheeks attached to our toothbrush, and they amplify DNA and match with the patient. Basically, the principle of polymerase chain reaction. There was a Nobel Prize given to Kerimolis, who designed this PCR in 1985 or so, based on the principle of DNA replication. So that's all for today's lecture. This is just telling you DNA as a one of the surprises we saw in last part of the last century about cloning sheep, the dolly, and that it's not only the egg cell and sperm cells, but using skin cell, the somatic cells, the 2N cells, its nucleus transplanted into enucleated oocyte of the donor mother. They could make a whole new sheep, which means the diploid nucleus of skin cell, it has the potential to give rise to a whole new organism, provided it is reprogrammed in such a way, and that reprogramming takes place within the oocyte because it has a very specialized cytoplasm. It can reprogram all the genome. Reprogramming means when this sheep was baby, the first cell was 2N fertilized egg, and 2N, through mitosis, 2N. Before it became the skin, now this skin cell has undergone a very, very specialized programming. It's more complex than your C++ or Python. This programming has switched on certain genes which are only responsible for making skin, not eyes, not liver, not heart, etc. So skin-specific genes are active in this cell. They are programmed. Now, if you want a skin cell to make a whole new baby, you have to reprogram. You have to what we call, you have to reset its software, and then resetting can only take place. They did this. They enucleated. They removed the nucleus of oocyte and placed the nucleus of what do we call that, skin cell, and this oocyte reprogrammed its software, and then a whole baby was made. And this is also in Jurassic Park. They tried to, mimic this principle. So please watch all this and note down your questions. All the time which you have for recitation as well as your lectures, this lecture is going to serve replacement of that time, and the lecture time will be just to answer your questions. Thank you very much.